We're part of the CHASM program, which looks at cancer. So we do a combination of oncogenomics, so that's kind of analyzing genomes to look for variants that drive cancer. But we also do a lot of functional genomics, so using CRISPR screens to identify which genes actually drive certain cancers. And the plan for today is for me to talk for about 10 minutes just to set the scene, give an idea of what we'll be doing over the course of the next 2 hours. And after that, we will break out into your groups, and then there'll be a collab notebook, the same way that you, similar to what you used yesterday with Jansu, with a self-directed exercise that contains some code for you to run, some plots for you to interpret, some questions to consider, and also, if you're someone who is more confident in coding, some opportunities to try out and solve some problems in R. Please don't feel like you need to be a confident coder to do this. A lot of this, the code is provided for you. My idea is that you take away the principles rather than the specifics of what the commands do. So without further ado, I will get started with setting the scene. So you've probably heard a bit about CRISPR screening already, so I won't take too long with this. But just so we're all on the same page as to what we'll be doing today, we're going to be looking at the kind of experiments where we deliver in a library of guide RNAs and Cas9 into a cell population, a pooled screen this will be. So this is in a single test tube, dish rather than test tube. And those guides are going to diversify our cell population in some way. That could be we use base editing to introduce mutations into the BRCA1 gene, or it could be we use genes targeting the entire genome to systematically delete every gene in the genome. And some of those modifications we make are going to have a deleterious effect on cell fitness if we're just doing an essentiality screen. So those cells will die and drop out from the screen. And what we're going to aim to do is identify those cells which are dropping out through sampling the population of cells at different time points. That might be at the start when we've initially transfected, and then at later days after cutting and loss of the gene has had a chance to manifest as an effect. And we're going to take the cell pellet samples that we get and submit them for high throughput sequencing, NGS, Illumina. You may be familiar with these words. And fundamentally, all that we're going to try and do is compare the counts of guides in different samples. So we'd expect that guide RNAs which deplete over the course of the screen represent genes that are essential. So we'll back infer from guide counts to genes for essentiality. And the things that we regularly use this for, that the community uses this for, are identifying hits. What are the genes that are crucial to some biological process? On the left here, I've got an example of a genome-wide screen where people knocked out all the genes in the genome to see which ones are important in SARS-CoV-2 getting into cells. And those guides which deplete tell us the proteins that are important to that viral entry mechanism. CRISPR screens get used a lot in cancer because we're interested in drug development and finding out which cancers are underpinned by what driving mutations. So which cancers are likely to respond to BRAF inhibitors. And the start point for the kind of analysis we'll be doing, so after the biology finishes, after you've prepared your cell pellets, chemistry has to happen. And once the chemistry's done, then we can do informatics and computational work. And we do a load of this here at Sanger, but I just want to make sure that we're all on the same page as to actually how we derive the data that we're going to be working with. So we start from fragments of DNA, and these are probably going to be your guide RNAs. So these might have been PCR'd and DNA cleanup, kind of stuff that is like a bit of a mystery to me, not really my side of the equation. But we're starting with what we call fragments of DNA, and we're rendering them amenable to sequencing by adding on adapter sequences. These just allow the fragments to anneal onto a surface within a sequencing machine, which we call a flow cell. The flow cell is patterned so that these sequences will anneal sparsely. There'll be large distances between them, which is going to be important for our ability to actually resolve between different fragments when we do the sequencing reaction. And then basically, if you're familiar with molecular biology, we're basically going to perform PCR reactions, but in a spatially localized manner. So given that the pieces of DNA we want to sequence are anchored by adapters, any PCR extensions will create child pieces of DNA which are located near to their parent. And through an iterative process of amplification, you end up with clusters of DNA sequences where each sequence in the cluster has the same base sequence. And this is important because it allows us to have, it's essentially like multiple replicates of a DNA sequence, which gives a strong signal in the reaction we use to call bases. And that reaction is one called sequencing by synthesis. You may be familiar with this. It's using fluorescently labeled dideoxy nucleotides, which we add iteratively onto a growing strand of DNA. And we use the colors that they contain to represent bases. So we end up with something that looks a bit like this. It's kind of like a night sky, or like pictures of the Hubble telescope wide field, where each of these spots represents a cluster of reads, and each of those spots also originates from a single fragment. So you can see there's many millions of these spots, and their evolution over time represents us iteratively figuring out which base is present at which position along the read. One thing to note is that this reaction gets more inefficient the longer that it goes on for. So we're often limited into how long of a region of DNA we can sequence with this method. Typically, it's about 150 base pairs. And that's all right for our case, because we'll be looking at guide RNAs, which are about 20 base pairs, 30 base pairs. So chemistry over now, we move on to the computational world. And that image of fluorescent labels that we took on the sequencing machine, we now deconvolute and turn into a DNA sequence. Based on the colors, we can infer what the base was, and for each position in that original flow cell, we can now figure out what the original DNA sequence that was there encoded, and we can also get some information about the metadata, so where in the flow cell that read originated in, what position on the XY axis it was, and we also get quality scores too. So these are how confident we are in a base call being accurate. So one thing that I mentioned in the last slide was that the sequencing by synthesis reaction decreases in efficiency over time, and that manifests in this kind of, over the millions of reads we have, most of them will have lower quality base calls at the end of the read. So you might want to trim off the end of a read and get the bit of DNA sequence that you're most interested in accurately identifying the middle of a read. So if any of you have ever used a FASTQ file or heard of a FASTQ file, that's a FASTQ file is essentially millions and millions of these 4 lines all pasted one after the other. And what we're going to start with today is counts data. So CRISPR counts data we get by processing these reads. So we might need to chop off extraneous bits of sequence. Maybe there was some PCR primers involved in the actual product that we sequenced. Maybe we need to reverse complement the read so that it matches what the guides we're expecting to look like are. So we'll do some pre-processing typically, and then we do a sort of matching stage where we look at our query read. So it's for every read in the millions that we generate, we compare it to the guides that we know should be in there and find which one it matches to. So here, my read is this purple bar, doesn't match to guide A, but it does match to guide B. So that's a tick for guide B, and we've got a count of 1 for guide B. We repeat this iteratively over a million reads, and we end up with a counts table that looks like this, where we have the guides in our library and counts for them. Now, we won't actually be doing any guide count generation in this practical because 1, it's quite slow, it's computationally intensive, and 2, it's actually quite straightforward to do normally. Normally, it's a single command where you provide your guide library, so the sequences of what the guides you expect to be in the library, and your sequencing file, so your FASTQ or your BAM or CRAM. Those are just ways of storing sequence data, and it will spit out whatever guide counting tool you use would spit out a counts table. If you're looking to do this guide counting for yourself and you don't have a friendly bioinformatician to hand, one tool I could recommend that's very straightforward to use is Magic Count. That's part of a very popular tool for CRISPR analysis. Another I could recommend is Luca, which is one we've developed here, which is much more flexible in terms of accommodating more complex designs to experiments. So if you want to do a dual guide experiment or a three guide experiment where you have three guides per plasmid, or if you want to use a different CRISPR system, we work a bit with Cas12 and Cas13, which have different PAM sites, different arrangements of where the protospacer is relative to the other parts of the guide. So this can support that kind of stuff too. So where we will be starting today is from a stage where we've generated all the counts for all of our samples that we're going to look at, and we'll have something called a counts matrix. So a counts matrix is quite a common object in bioinformatics. It's a table basically where you have your samples as columns. So samples might be time points, they might be drug treatment conditions, they might be replicates, and you have your guides as rows. This could also be transcripts in transcriptomics or species in a metagenomic study. So there's quite a lot of overlap between the methods we'll be looking at today and different kinds of bioinformatics analysis. So I'm hoping that you might take away some general principles too for the future. What's kind of unique about this sort of counts matrixy data is that we'd call it high dimensional. So we have a limited number of columns, but we have loads of rows because we've got loads of guides. Typically, we're looking at like 80,000, 100,000 guides per library. And the toolkit that we use to analyze them often involves doing some initial exploratory analysis of our data and QC, some transformation to render that data suitable for doing comparisons, and then associations testing. So finding out what your hits are, doing the actual interesting bit of this gene is important to this process. And that's what we'll be basically walking through today. To run through these at a high level, one thing we'll be doing is normalizing our data set. So you may have normalized, if you're someone who works in the lab, you may have normalized DNA concentrations. Normalization is something that we have to apply to in the world of bioinformatics. They're just families of methods that allow us to render samples comparable. We'll be doing a very simple normalization method here where we just try and bring the medians of guide counts into alignment across samples. And this helps you fix things. Imagine you had submitted one sample with twice the amount of DNA with all the others, and you got 2 million reads for sample A versus 1 million reads in all the other samples. Normalization will allow you to find some modification to the read counts from sample A so that you can compare it with sample B and C, etc. So we'll get raw counts from our counting software, normalize that data, and then we'll proceed to what's called associations testing. This is like the statistics bit. By associations testing, I mean finding X which is associated with Y. So that might be, in our case, guides that are associated with the day 18 time point. That could be proteins associated with drug response. So association is where we do our statistical testing. And I'm sure many of you will have performed simple statistical tests like a student's t-test, where we take the mean of one sample and mean of one condition and try and compare it to the mean of another. We're basically going to be doing something similar here. We're going to compare the mean of guide counts in our initial condition versus the end of our screen to find which ones are statistically significant. The only difference here is that where students' t-test assume some stuff about normal distributions, we have to make some modifications to that, so apply some different statistics because we're dealing with counts data and that's not normally distributed. And the other thing we have to bear in mind is that we're going to be doing a large number of comparisons. So if we're doing 80,000 t-tests, then you would get a lot of false positives. And it would be kind of a p-hacking exercise. So we use statistical correction methods to limit the false positives that we'll get out as guides that are important to cellular fitness. To just dwell a little bit on the mechanics of this, we will be looking at changes expressed as log fold changes. So this is quite common in bioinformatics, is rather than doing linear comparisons, we do things on an exponential logarithmic scale. So this guide A has a mean count of 100 in day 0 and a mean count of 200 at day 18. So it's effectively doubled in counts in this example. We'd represent that as a log 2-fold change, which is equal to 1. So it's kind of a 2 to the power is what we're using. So if you do 2 to the power 1, then you get 2. I think, yeah, you do. So that's a doubling of counts. 2 to the power 0 is 1. So that's a count stay the same. So when you're looking at log fold change values, bear in mind that we're operating on a distorted and weird scale. And our association tests are going to try and find what are the interesting guides here. Guide A looks quite interesting to me because it doubles. Guide B doesn't look interesting. It doesn't change over. Guide D maybe looks interesting because there's a change in the means, but I've simulated this as there's quite a lot of variability between replicates counts of those guides. So we need a statistical method that can weigh what the spread of the data is in each condition. So we're almost ready to go. The exercise in this practical is we're going to start with a counts matrix, and we're going to use it to identify hits. So our hits will be genes that are essential in the HAP1 cell line. If you've not come across it before, HAP1 is a sort of workhorse in gene editing and CRISPR experiments because it has this unique property of having it's haploid. It's a human cell line, but it only has one copy of each chromosome, which means that you can avoid all sorts of annoying things like mixed penetrance of genome editing. So that makes it really handy. And the structure of what we'll be doing is we have a Colab notebook. Can you all see my Colab notebook? Can I get a thumbs up if that is visible to you all? So as I mentioned earlier, this has some text walking you through things, and it's got code cells for you to execute. Install several things, and there may be steps where you have to wait for a minute for something to run because these things aren't instantaneous. But normally you can run your cells by clicking the play button or by if you hover over, you should get a message as to how to actually trigger them. I use a Mac, so mine is Control Enter. And once a cell has run, you can then proceed to running cells beneath them. So now that cell has a tick next to it. I know I'm ready to move on to the next stage. While you're waiting for commands to run, I'd encourage you to read above and below them, just in case there's pertinent information to the task. Often if something hasn't worked, it might be because there's an explanation above or below it. And without further ado, I think I'll probably set you all loose. So I will post a link to the GitHub page that this is on in the chat, and I'll just demo how to open the Colab notebook as a reminder from yesterday. Yeah. So CRISPR counts analysis is going to be the one that we're working from today. And you can hopefully see that in my GitHub here. There should be a big blue opening Colab button, which if you press, should take you through to a copy of the notebook that I'm working in.
I was just checking the other tool, GS screen. So it says it works similar as Magic in R, but for simple screens?
Yeah, so there are CRISPR screens which just involve the depletion of essential genes, but you could imagine a more complex experimental design where you had multiple factors. So if anyone's ever done a transcriptome, like everyone's ever used DSeq for doing RNA analysis, you can have multiple factors in a model. So you could incorporate the effect of a drug and the effect of cell lines and try to tease out interactions between different experimental factors. So if you're just looking at condition A versus condition B, then both Magic and Bagel are very good for that in the way we've explored. If you're looking at condition A versus B versus C with a bit of extra stuff on top, then you want something like DESeq from the RNA world, or you can use Magic in a mode called MLE, maximum likelihood estimation.
Yeah. To follow up on that, because when we talk about the depletion of the essential genes, even at time zero, there's some depletion going happening also at time zero. So
Yeah,
from the time zero, let's say I collected my extraction, the genomic DNA for sequencing, and then my conditions, two controls, two treatments. Even so, when we look at the comparison of treatments, let's say after 5 days, T5, so should I be comparing my treatment versus untreated at time 5, or I should be comparing my treatment with time 0? Or I can do both, or there is no
you can do both. So typically you're going to generate a library of DNA that you're going to input to the cells.
Mhm.
And you then deliver that in via a lentivirus.
Yeah,
So
I did that.
you can just sequence that input. You can take that library, prepare it for DNA sequencing, and submit that for DNA sequencing. And in many ways, what that point is trying to get at is that that's often the best control, because when you put stuff into cells, um, even your time point zero normally is the cells have to recover from transfection. So you
Mhm.
might collect them a day after you did the transfector, and even that is enough time for Cas9 and the guide to start cutting and depleting them. So the point of that is if you can use a plasmid library as a control for doing your comparisons too, then that's the best one to use.
Oh, okay, okay. So it's context specific what I am going to look into. So there's not a general rule, right? Yeah,
No, I don't think so.
yeah. Because when I did comparison of my untreated and the treated versus time 0, obviously the number of guide are in abundance and the results were different.
Mhm.
Yeah.
Anyone want to offer any more questions, or you've got me? I've got a question in the chat. Jamie Billington, do you think Magic could be suitable for the analysis of sorted samples from a crisp pooled screen? So is this talking about having flow sorted samples?
Yeah, I'm just meaning like as a end time point, indeed to have sorted samples to compare, and maybe to have these samples of different sizes. So maybe one sample with 1 million cells and then the other with 10 million cells. So if
Yeah,
Magic is suitable.
I have actually done this on flow sorted cells. It works okay, is the answer. And normally the thing you want is to make sure you collect enough cells in your sorting, as many cells as possible in your sorting, because if it impacts the coverage, if you can only get a small fraction of cells. There is a tool called WaterBear, which is sort of designed for comparing across flow sorted cell populations for doing CRISPR analysis. It assumes that you are like sampling all of the cells, but you're just binning them depending on where they sit in the flow spectrum. But Magic's worked fine for that when I've done like the top. We needed to do. We had an antibody marker and looking at expression of a cell surface protein. So we needed to compare the low fluorescent cells with the high fluorescent cells, and that worked fine. But it was very noisy actually because of just the low number of cells that you get out of sorting um, in that particular experiment.
May you please type the name of this other tool that you suggested, please?
Absolutely, yeah.
Thanks.
I'll send you a link to the paper.
I have a question. It's a bit of follow-up to Ilaria's question, if that's possible. It's just if you want to do the same thing, it's not instead of using a fax sorting, you would use like a transwell where you have the cells going through a membrane. Then you're not sure of how many cells you get underneath, like on the bottom part. That would have a variant number of cells from sample to another. Would that be the same thing to use to analyze it, or what's the as well the coverage that I would aim for?
It's difficult for me to give you a coverage estimate
Yeah.
without knowing lots of details about the
Yeah.
experiment. I as many cells as possible.
Okay. Would be
And
500 coverage would be fine. I've done 1,000 for a normal one in a plate, but like on a
I suspect that would be all right.
Yeah.
But I can't promise these
Okay.
experiments will work. I don't want
No
to be
worries.
accountable. Again, I think magic would probably work in the situation that
Okay.
you're talking about.
Great. I have to say that the part that we did on Google Colab was very well explained, and I really appreciate that, because for me as not a non-mathematician, I'm pure biologist. That was very clear, and each part was explained very well. That was very good. Thank you.
Really
I have
positive.
tried the other part where the last question says optional if you are confident. I'm not confident in R. I have to admit that I've used the Gemini part here on Google Colab.
I see.
It generated me a what's called now like a code here that I plotted, and it used the scatter plot. Do you think that's a good one, or is there other ways of plotting this type of data?
So this is in, I think this is an answer to one of the last questions, which was I asked
to compare Bagel and Magic.
the Bagel and the magic outputs. So
Yeah.
I'd say one way is to do this is with a scatter plot where you look at the correlation between them. That's probably what I would have done, because I'd be interested in which genes differ between them. So you'd expect genes that have similar scores to fall along a line.
Yeah.
The outliers to appear either side of that line. But you could also, I think a Venn diagram would be quite helpful here too.
Okay.
To assess the degree of overlap between sample A and sample B, between method A and method B.
Okay. Thank you so much. Thank you, Jamie.
Thank you so much for your time. I've really enjoyed today. And thank you, Simon, too, for your help. Much appreciated.